Imagine, for a moment, an ocean 500 million years ago. The water is warm and shallow, lit by sunlight filtering through layers of algae and drifting particles. The seafloor is a different world entirely from what we know today. Here, soft-bodied creatures glide and pulse through the currents, vulnerable, exposed, their lives defined by the constant threat of being consumed. There are no shells yet, no armour, no spines, just flesh, moving carefully through a world that has begun to change. Then, in what seems like an instant in geological time, everything shifts. Hard parts appear. Exoskeletons emerge from what was once only soft tissue. Calcium carbonate plates form where there was once just skin. The first protective coverings in the animal kingdom begin to take shape, and with them, life on Earth transforms forever. This is where our story begins tonight, in those ancient seas, at the very moment when animals discovered how to build walls around themselves. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. Tonight, we're stepping into one of evolution's most remarkable innovations, the development of shells, armour, and protective structures. We'll journey from the earliest calcified tubes in ancient oceans to the intricate exoskeletons of insects, from the bony plates of armadillos to the shock-absorbing spines of hedgehogs. Along the way, we'll discover how life learned to build barriers between itself and danger, how calcium and chitin became the building blocks of survival, and how the simple act of growing a shell changed the course of life on Earth. So settle in, let your thoughts drift, and prepare to explore the evolutionary story of protection itself. Over 540 million years ago, during a period geologists call the Cambrian, something extraordinary happened in Earth's oceans. Before this time, most life consisted of simple, soft-bodied organisms. Sponges filtered water through their porous bodies. Worms burrowed through sediment. Jellyfish-like creatures pulsed lazily through the currents. These animals had no hard parts, no shells, no armour to speak of. They existed in a world where survival meant staying hidden, moving slowly, avoiding attention. Then, in a span of perhaps 20 to 25 million years, which sounds like an eternity to us, but represents merely a heartbeat in the 4.5 billion year history of our planet, life exploded into complexity. Scientists call this the Cambrian explosion, and it marks one of the most significant turning points in the history of life. During this time, nearly all major animal body plans that exist today first appeared in the fossil record. Creatures with segmented bodies emerged. Animals with jointed legs scuttled across the seafloor. And most significantly for our story tonight, animals began building hard protective structures for the first time. The catalyst for this transformation remains a subject of intense scientific debate. But many researchers believe that rising oxygen levels in the ocean played a crucial role. Oxygen is the fuel that powers complex life. It enables the production of collagen, the protein that holds animal bodies together. It allows for more sophisticated metabolism, supporting larger bodies and more active lifestyles. As oxygen levels climbed, they crossed a threshold that made it energetically feasible for animals to construct hard parts, to invest the enormous resources required to build shells and exoskeletons. But there was another factor at play, one that would drive the evolution of armor for hundreds of millions of years to come: predation. The Cambrian seas witnessed the emergence of the first true predators, animals that actively hunted and consumed other animals. One of the most fearsome was Anomalocaris, a creature that could reach lengths of up to 1 meter. It possessed large, sophisticated eyes mounted on stalks, allowing it to spot prey from a distance. Its mouth was a circular arrangement of sharp plates, and it used 2 grasping appendages to capture and manipulate its victims. The appearance of such predators transformed the ocean from a relatively peaceful place into an arena of constant evolutionary competition. This is where the arms race began. Prey species that could develop defensive adaptations, anything that made them harder to catch or less pleasant to eat, had a better chance of surviving long enough to reproduce. Hard shells offered protection against crushing jaws. Spines made animals difficult to swallow. Thick exoskeletons could withstand attacks that would have been fatal to soft-bodied creatures. Natural selection favored those innovations, and over countless generations, protective structures became more sophisticated, more diverse, more integral to survival. Among the most successful of these early armored animals were the trilobites. These marine arthropods first appeared in the fossil record around 521 million years ago, and they would dominate ocean ecosystems for nearly 300 million years. Trilobites had segmented bodies divided into 3 lobes, which gave them their name. Their bodies were covered in a hard exoskeleton made of calcite, the same mineral that forms limestone and chalk. This calcified covering provided excellent protection, while still allowing for flexibility and movement. The trilobites' success wasn't just about armor. They also developed compound eyes, some of the first complex visual organs in the animal kingdom. These eyes, made of calcite crystals arranged in precise geometric patterns, allowed trilobites to see their world with remarkable clarity. They could detect predators approaching, spot food sources, and navigate their environment with a sophistication that gave them a tremendous advantage. Over the course of their long evolutionary history, trilobites diversified into thousands of species, some as small as a millimeter, others growing to lengths of over half a meter. Some developed elaborate spines and horns, likely as additional defensive adaptations. Others had smooth, streamlined bodies built for burrowing. What makes trilobites particularly significant to our understanding of early armor is that their fossilized remains are incredibly abundant. Because they possessed hard exoskeletons, they preserved well in sedimentary rock, leaving behind a detailed record of their existence. When we examine these fossils, we can trace the evolution of their protective structures, seeing how different species adapted their armor to different environments and different threats. We can observe the gradual refinement of their defenses, the way evolution tinkered with their body plans over millions of years, always favoring those innovations that improved survival. The Cambrian explosion established patterns that would persist throughout the history of life. The emergence of predators created selective pressure for defensive adaptations. Those adaptations, in turn, created pressure for predators to evolve better hunting strategies. This back and forth, this endless cycle of adaptation and counter-adaptation, drove much of the diversification we see in the animal kingdom. Even today, hundreds of millions of years later, the predator-prey arms race continues to shape evolution, and the protective structures that first appeared in those ancient Cambrian seas remain fundamental to how animals survive. When you hold a seashell in your hand, you're holding one of nature's most elegant architectural achievements. That smooth, spiraling structure, with its iridescent inner surface and tough outer layer, represents millions of years of evolutionary refinement. But shells are more than just beautiful objects washed up on beaches. They're sophisticated biological structures, grown by animals through a remarkable process called biomineralization, and understanding how they form reveals one of evolution's most ingenious solutions to the problem of protection. Shells are produced primarily by mollusks, a phylum that includes snails, clams, oysters, and numerous other species. These animals possess a specialized tissue called the mantle, which lies just beneath the shell and is responsible for its construction. The mantle is lined with specialized cells that secrete both organic proteins and mineral compounds. These materials are released into a narrow space between the mantle and the existing shell, where they combine to form new shell material. Think of it as laying down steel reinforcement bars and then pouring concrete over them. Except the process happens at a microscopic scale, molecule by molecule, layer by layer. The primary mineral in most shells is calcium carbonate, the same compound found in chalk, marble, and coral. Mollusks obtain calcium from their environment, absorbing it through their gills, digestive system, and epithelial tissues. This calcium is transported through the animal's body by its hemolymph, which is essentially its blood, and delivered to the mantle tissue. There, specialized cells store the calcium in tiny granules until it's needed for shell construction. But calcium carbonate alone wouldn't create the strong, resilient structure of a shell. The secret lies in how that calcium carbonate is organized, and that organization is controlled by proteins secreted by the mantle. These proteins form a framework, a scaffold upon which calcium carbonate crystals can grow. Different proteins cause the calcium carbonate to crystallize in different forms. The two main forms found in shells are called calcite and aragonite. They're both calcium carbonate, but the calcium and carbonate ions are arranged in different geometric patterns, which gives them distinct properties. Calcite tends to form in more blocky cubic arrangements and is generally stronger under compression. Aragonite has a different crystal structure, more needle-like, which makes it slightly more flexible. Many shells use both forms, deploying each in different layers where its specific properties are most advantageous. The proteins don't just determine which form of calcium carbonate develops. They also control when and where crystals start growing, how fast they expand, and when they stop. This precise regulation allows mollusks to create shells with incredibly complex internal architectures. A typical mollusk shell has 3 distinct layers, each with its own function. The outermost layer, called the periostracum, is primarily made of protein. It's often rough in texture and may have bumps, ridges, or even spikes. This layer serves as the first line of defense against abrasion and helps protect the underlying layers from dissolving in acidic water. Some species shed this layer as they grow, while others maintain it throughout their lives. Beneath the periostracum lies the prismatic layer, where calcium carbonate crystallizes in a form that creates a tough, rigid structure. The proteins in this layer cause the calcium carbonate to form calcite crystals that fit together like bricks in a wall. These calcite prisms fill in the spaces between protein fibers, creating a composite material that's remarkably resistant to cracking. This layer provides much of the shell's structural strength, the hardness that makes it difficult for predators to crush or penetrate. The innermost layer, the one in contact with the mantle, is called the nacre, or mother of pearl. This is where the shell's iridescent beauty comes from. Nacre consists of thin platelets of aragonite crystals, arranged in layers, separated by sheets of protein. The thickness of these layers is precisely controlled, often measuring just a few hundred nanometers. When light hits these layers, it reflects and refracts, creating interference patterns that produce the shimmering, rainbow-like colors we associate with the inside of shells. But nacre isn't just beautiful, it's also incredibly tough. The layered structure allows it to absorb impacts by deflecting cracks along the protein sheets, preventing catastrophic failure. As a mollusk grows, its shell must grow with it. This happens along the shell's outer edges. A snail, for instance, adds new material around the opening where it extends its body. For a clam or oyster, growth occurs along the margin where the two halves of the shell meet. This incremental addition creates growth rings, similar to the rings in a tree trunk. Scientists can examine these rings to determine a mollusk's age and even reconstruct the environmental conditions it experienced during its lifetime, because the chemistry of the shell material reflects the chemistry of the water in which it formed. The evolution of shell building represents one of the earliest examples of biomineralization in the animal kingdom. Fossils of early shell-bearing organisms appear near the end of the Ediacaran period, just before the Cambrian explosion. These first shells were simple tubes or cone-shaped structures, but they represented a profound innovation. By sequestering calcium carbonate from seawater and organizing it into a protective covering, these early animals gained a significant advantage. They were harder to eat, more resistant to environmental stresses, and able to support larger body sizes. Over time, different mollusk lineages evolved shells suited to their particular lifestyles. Snails developed coiling spiral shells that efficiently enclosed their organs while remaining relatively lightweight. Bivalves like clams created two hinged shells that could close tightly, sealing the animal inside when danger threatened. Cephalopods, the group that includes octopuses and squids, evolved in a different direction. Their ancestors had external shells, but most modern cephalopods have either reduced their shells to small internal structures or lost them entirely, trading protection for the agility needed for their active, predatory lifestyles. The chemistry of shell formation is intimately tied to ocean chemistry. The balance of calcium and carbonate ions in seawater, the ocean's pH, and its temperature all affect how easily shells can form. Throughout Earth's history, these conditions have fluctuated, sometimes favoring organisms that build shells from calcite, other times favoring those that use aragonite. These shifts have influenced which shell-building species thrived and which struggled. Today, as atmospheric carbon dioxide dissolves in the ocean and makes seawater more acidic, many shell-building organisms face new challenges. The increased acidity makes it harder for them to extract calcium carbonate from the water and to prevent their shells from dissolving. This is one of the quieter consequences of climate change, but it has profound implications for marine ecosystems. If you've ever examined a crab's shell or held a cicada's molted exoskeleton, you've encountered another of evolution's great protective innovations, the arthropod cuticle. This isn't made of calcium carbonate like mollusk shells. Instead, it's built from an entirely different material, a polysaccharide called chitin, combined with proteins and sometimes reinforced with minerals. Chitin is one of the most abundant organic materials on Earth, forming the structural framework for the exoskeletons of insects, spiders, crustaceans, and countless other arthropods. Understanding how this material works reveals a different approach to the problem of armor, one that emphasizes flexibility and lightness alongside strength. Chitin is a long-chain polymer, structurally similar to cellulose, the material that makes up plant cell walls. It's synthesized within the epidermis, the outer layer of an arthropod's skin, from sugars and amino sugars. These long molecular chains are organized into fibers, which are then embedded in a matrix of proteins. The result is a composite material, something like fiberglass, where the chitin fibers provide tensile strength, while the protein matrix binds everything together and allows some degree of flexibility. In its unmodified form, chitin is translucent, tough, and somewhat pliable. You can see this in the soft cuticle of many caterpillars and other insect larvae, where the exoskeleton needs to be flexible enough to allow for growth and movement. But most arthropods modify their chitin through additional processes that dramatically alter its properties. One of the most important modifications is called sclerotization, which involves cross-linking the protein molecules in the cuticle. This cross-linking hardens the material, making it much stiffer and more resistant to deformation. Different parts of an arthropod's body undergo different degrees of sclerotization, creating hard plates where protection is needed, and flexible membranes at joints where movement must occur. Some arthropods, particularly crustaceans like crabs and lobsters, take this a step further by mineralizing their exoskeletons. They incorporate calcium carbonate into the chitin protein matrix, often in concentrations so high that the mineral makes up more than 95% of the dry weight of the cuticle. This biomineralization creates armor that's extremely hard and resistant to crushing. Interestingly, crustaceans don't use the same minerals everywhere in their bodies. The main body armor might be heavily calcified, while the joints remain more flexible. The parts that need to be particularly hard, like the tips of claws or the cutting edges of mandibles, are often reinforced with extra layers of sclerotized protein, which is actually harder than calcite. The structure of arthropod cuticle is hierarchically organized, with complexity at multiple scales. At the molecular level, chitin chains align parallel to each other, forming crystalline regions. These chitin crystals are embedded in the protein matrix. At a larger scale, the chitin protein fibers are arranged in layers. In many arthropods, these layers have a distinctive pattern. The fibers in one layer run parallel to each other, but each successive layer is rotated by a small angle relative to the one below it. This creates a twisted plywood structure called a helicoidal arrangement, which gives the exoskeleton exceptional strength in all directions and helps prevent cracks from propagating. The outer surface of an arthropod's exoskeleton is coated with a thin layer called the epicuticle, which is primarily composed of lipids and proteins. This layer is crucial for terrestrial arthropods because it prevents water loss. The waxy components of the epicuticle create a waterproof barrier, allowing insects and other land-dwelling arthropods to maintain their internal fluid balance even in dry environments. Without this protective coating, arthropods would quickly desiccate, limiting them to aquatic or very humid habitats. But there's a significant trade-off to having an exoskeleton. It doesn't grow with the animal. Once the cuticle hardens, it becomes a rigid shell that cannot expand. For an arthropod to increase in size, it must periodically shed its entire exoskeleton in a process called molting or ecdysis. This is an elaborate and risky undertaking. Before molting, the animal secretes enzymes that digest much of the inner layers of its old cuticle, recycling the materials. Simultaneously, a new, larger cuticle forms underneath the old one. When the time comes, the old exoskeleton splits along predetermined lines, and the arthropod pulls itself out, soft and vulnerable. During this brief window, before the new exoskeleton hardens, the arthropod is at its most defenseless. It cannot effectively flee from predators. Its soft body offers no protection against attacks. Many arthropods hide during this period, sometimes for days, remaining motionless until their new armor has hardened sufficiently. To expand the new exoskeleton before it hardens, they often take in water or air, swelling their bodies to stretch the pliable cuticle. Once the cuticle has hardened through tanning and mineralization, the arthropod is larger, but it won't be able to grow again until the next molt. The frequency of molting varies among species and changes over an individual's lifetime. Young arthropods molt frequently, sometimes every few weeks, as they grow rapidly. As they mature, molts become less frequent. Some arthropods, like many insects, stop molting entirely once they reach adulthood. Others, like lobsters and many other crustaceans, continue molting throughout their lives, which is why some individuals can reach extraordinary sizes if they live long enough and have access to sufficient food. The evolution of the arthropod exoskeleton was a pivotal moment in the history of life. It appeared early, likely during or before the Cambrian explosion, and it provided arthropods with both protection and structural support. Unlike animals with internal skeletons, arthropods could attach their muscles directly to their exoskeleton, creating efficient lever systems for movement. This combination of protection, support, and mobility helped make arthropods the most successful group of animals on Earth. Today, they represent more than 80% of all known animal species, inhabiting virtually every environment from the deepest ocean trenches to high mountain peaks. Among all the protective structures in the animal kingdom, perhaps none is more unusual than the turtle shell. This isn't an exoskeleton that the animal builds and periodically sheds. It isn't a structure secreted by specialized tissues like a mollusk's shell. Instead, the turtle shell is part of the animal's skeleton, a fusion of modified bones that encloses the turtle's body in a rigid dome of protection. Understanding how this structure evolved and how it develops reveals one of evolution's most radical redesigns of the vertebrate body plan. The shell consists of two main parts: the carapace, the domed upper shell, and the plastron, the flatter lower shell. These two sections are connected by bridges of bone on each side of the turtle's body, creating a box that protects all of the animal's vital organs. What makes this structure so remarkable is that the carapace is formed from the turtle's ribs and vertebrae. During embryonic development, the turtle's ribs grow outward, extending laterally instead of curving downward as they do in other vertebrates. These broadened ribs then fuse with dermal bone, bone that forms in the skin, creating the composite plates that make up the shell. This is a profound departure from the typical vertebrate body plan. In most animals, The ribs form a cage that protects the heart and lungs, with the shoulder blades positioned outside of that cage. But in turtles, the process of rib extension and fusion traps the shoulder blades inside the rib cage. This unique arrangement is found nowhere else in the animal kingdom. It required significant modifications to how the turtle's skeleton develops and how its muscles attach and function. Scientists studying turtle embryology have identified a specialized structure called the carapacial ridge, which appears early in development and initiates the sequence of events that leads to shell formation. This ridge acts as a signaling center, releasing chemical signals that cause the ribs to grow outward and the shoulder girdle to reposition itself. The individual plates that make up the carapace are called costals when they're formed from ribs, and neurals when they're formed from vertebrae. Around the edge of the carapace, a series of peripheral plates, which are purely dermal bone not attached to the ribs or vertebrae, complete the dome. All of these bony plates are covered by a layer of large epidermal scales called scutes, which are made of keratin, the same protein that forms our fingernails and hair. These scutes don't align with the underlying bones. Instead, they overlap the seams between bones, creating a pattern that strengthens the shell and helps prevent cracks from spreading. The plastron, the lower shell, develops through a different process. It's formed entirely from dermal bone, with no contribution from the animal's ribs or vertebrae. Scientists believe that the plastron may be derived from the gastralia, paired floating ribs that were present in many early reptiles, and that still exist in some modern reptiles like crocodiles. In turtles, these bones expanded and fused to create a solid protective shield for the animal's belly. Fossil evidence shows that the evolution of the turtle shell didn't happen all at once. Paleontologists have discovered several species of ancient turtle ancestors that possessed partial shells. One remarkable fossil, Odontochelys semitestacea, which lived about 220 million years ago during the late Triassic period, had a fully formed plastron, but no carapace. Its ribs were broadened and T-shaped in cross-section, suggesting they were in an intermediate stage of development. But they hadn't yet fused into a complete shell. Another fossil, Papachelys, is even older, dating to about 240 million years ago. It had broadened ribs and the beginnings of a plastron, but both structures were far from complete. These transitional forms reveal that the turtle shell evolved gradually, likely beginning with broadened ribs that provided some protection and possibly aided in burrowing, a behavior that may have helped early turtle ancestors survive harsh environmental conditions. Over time, natural selection favored increasingly complete protection, eventually leading to the fully enclosed shell we see in modern turtles. This evolution required numerous genetic and developmental changes, including modifications to regulatory genes that control body patterning. Researchers have found that many of the genes involved in shell development are ancient genes that exist in other vertebrates but are expressed in new locations and at new times in turtle embryos, a process called evolutionary co-option. While the turtle shell provides extraordinary protection, it comes with significant costs. The rigid structure limits flexibility, making some movements difficult or impossible. Most turtles cannot use their ribs for breathing, as other reptiles do. Instead, they've evolved alternative breathing mechanisms, using muscles in their limbs and shoulders to pump air in and out of their lungs. The shell also affects thermoregulation. Because it's relatively thick and insulated, turtles cannot efficiently exchange heat through their shells. They rely instead on behavioral strategies, basking in the sun to warm up or seeking shade and water to cool down. Different turtle species have evolved variations on the basic shell design, adapting it to their particular lifestyles. Aquatic turtles often have relatively smooth, streamlined shells that reduce drag as they swim. Terrestrial tortoises tend to have higher, more domed shells, which provide excellent protection against predators. Some species have developed hinges in their shells, allowing them to close the front or rear openings completely, creating an impenetrable fortress. Sea turtles have reduced the weight of their shells, making them lighter and more hydrodynamic, though this makes them somewhat less protective. A few species, like the soft-shelled turtles, have greatly reduced the ossification of their shells, leaving them with a leathery covering that's much more flexible, but offers less defense. The turtle shell stands as a testament to evolution's ability to radically transform existing structures to serve new functions. By co-opting the ribs and vertebrae, structures that originally evolved for support and protection of internal organs, turtles created one of the most effective suits of armor in the animal kingdom. This innovation has served them well. Turtles first appeared more than 200 million years ago, and they've survived multiple mass extinction events that wiped out many other groups, including the non-avian dinosaurs. Today, they inhabit environments ranging from deserts to oceans, from tropical rivers to temperate woodlands, their shells providing them with protection that has proven effective across hundreds of millions of years. Not all protective structures are built from the bones of an animal's skeleton. Some emerge from the skin itself, forming a different kind of armor called dermal ossifications, or osteoderms. These are bony deposits that develop within the dermis, the deeper layer of the skin, creating scales, plates, or other structures that serve as protective covering. Among mammals, this type of armor is rare, but where it does occur, it's spectacularly effective. The most familiar example is the armadillo, whose name literally means little armored one in Spanish, and whose bony carapace represents one of the most distinctive forms of mammalian protection. Osteoderms form through a process distinct from the formation of regular bones. They develop from specialized cells within the skin's connective tissue, without any connection to the animal's internal skeleton. As these cells differentiate and begin producing bone tissue, they create small, individual bony plates embedded in the skin. Over time, these plates can grow larger and may develop intricate surface textures. In armadillos, hundreds or even thousands of these individual osteoderms fit together like tiles in a mosaic, connected by flexible collagen fibers and covered by a layer of tough, keratinous scales called scutes. The armadillo's carapace consists of three main sections: a solid shield over the shoulders, a solid shield over the hips, and a series of flexible bands across the midsection. The number of bands varies by species and gives some armadillo species their names. The 9-banded armadillo, the most widespread species and the only one commonly found in the United States, typically has 9 movable bands, though the number can range from 7 to 11. These bands are crucial because they allow the otherwise rigid armor to flex, giving the armadillo the ability to move, burrow, and in some species, curl into a ball. Each osteoderm is a complex structure. The bone itself is highly vascularized, meaning it contains many blood vessels that supply nutrients and remove waste. It's also innervated, connected to nerve endings that provide the animal with sensation, allowing it to feel touch and pressure through its armor. This makes the armadillo's carapace a living, dynamic structure rather than a dead shell. The surface of each osteoderm is marked by grooves and pits that correspond to the attachment points for the keratinous scutes above and the hair follicles and glands within the skin. These surface markings are so distinctive that paleontologists can identify armadillo species from isolated osteoderms, even millions of years after the animal's death. The evolution of osteoderms in mammals is particularly interesting because it represents a reacquisition of a trait. Early reptiles possessed extensive dermal armor, and many dinosaurs, including the famous ankylosaurs and stegosaurs, had elaborate osteoderm-based defenses. But when the mammalian lineage split from other amniotes more than 300 million years ago, the ancestors of mammals largely lost this trait, developing instead a covering of fur and a more active, metabolically intensive lifestyle. For osteoderms to reappear in groups like armadillos, the genetic pathways that control their development had to be reactivated. Recent research suggests that the genetic machinery for making osteoderms may never have been completely lost. Instead, it appears that the genes and developmental processes involved in forming dermal bone have deep evolutionary roots shared across many vertebrate lineages. Under the right selective pressures, when armor becomes advantageous for survival, these ancient pathways can be reactivated. For armadillos and their extinct relatives, the glyptodonts and pamphotheres, living in environments with predators and competing for food in semi-arid and grassland habitats, the benefits of armor outweighed its costs. Glyptodonts, in particular, took the armadillo body plan to an extreme. These extinct relatives of modern armadillos, which lived until about 10,000 years ago, could reach the size of a small car, with some species growing to nearly 4 meters in length. Their carapaces were massive, dome-like structures composed of thousands of osteoderms, fused into a solid, rigid shell with no flexible bands. Some glyptodonts also possessed heavily armored tails, which ended in spiked clubs that could have been used as weapons. These creatures were living tanks, so heavily protected that few predators would have dared to attack them. Yet despite their impressive defenses, they went extinct at the end of the Pleistocene, possibly due to climate change, habitat loss, or overhunting by early humans. The development of osteoderms involves a finely balanced interplay of genetic signals. During embryonic development, cells in the dermis receive chemical signals that cause them to differentiate into bone-forming cells. These signals activate genes that control the production of bone matrix proteins and the deposition of minerals. The timing and location of these signals determine where osteoderms will form and how they'll be shaped. In armadillos, this process doesn't begin until after birth. Young armadillos are born with soft, leathery skin. The osteoderms begin forming within a few weeks and gradually harden over the first months of life, eventually creating the rigid armor that will protect the adult. Interestingly, osteoderms have evolved independently many times across different animal groups. Besides armadillos, they're found in crocodiles, where they form the scutes on their backs and sides. Many lizards have osteoderms, particularly species like skinks and some geckos. Extinct dinosaurs possessed elaborate osteoderm-based armour. Even some modern rodents, such as the spiny mice of Africa and the Middle East, have recently been discovered to have small osteoderms in their tails. This pattern of repeated, independent evolution suggests that the developmental pathways for creating dermal bone are readily available to animals that need them, waiting to be activated when the selective pressure for armour becomes strong enough. The scutes that cover osteoderms serve multiple functions beyond simply providing an outer coating. They're made of keratin, arranged in layers that are continuously renewed as the outer layers wear away. This creates a self-renewing protective surface that resists abrasion and helps keep the underlying bone from being damaged. The scutes also provide additional sites for sensory organs, allowing the animal to detect touch and vibration through its armour. In armadillos, sparse hairs grow between the scutes, penetrating through the armour to provide another sensory modality. The combination of bone and keratin creates a composite structure that's tougher than either material alone, a principle that evolution has discovered independently in many lineages. When you think of protective adaptations in mammals, armour might not be the first thing that comes to mind. But there's another form of defense that's equally effective, and perhaps more widespread: spines and quills. These modified hairs have evolved in several distinct mammalian lineages, including hedgehogs, porcupines, echidnas, and tenrecs. Each group developed spines independently, and each has adapted them to serve slightly different functions. Yet despite their independent origins, there are remarkable similarities in how these structures work, offering a clear example of convergent evolution, where similar solutions arise to similar problems. Hedgehogs are among the most familiar spiny mammals, their backs covered with thousands of short, stiff spines. These spines are hollow hairs, stiffened by keratin and reinforced by an internal structure of longitudinal and transverse supports. Each spine is about 2 to 3 centimeters long, and an average hedgehog carries 5,000 to 7,000 of them across its back and sides. Unlike the quills of porcupines, hedgehog spines don't detach easily. They're firmly anchored in the skin, held in place by a bulbous base that prevents them from pulling out. When a hedgehog feels threatened, it contracts powerful muscles that pull its spiny skin over its head and body, while simultaneously curling into a tight ball. This draws all of its vulnerable parts, its face, belly, legs, and tail, into the center of a protective sphere. The spines stand erect, pointing outward in all directions, creating an impenetrable barrier. The muscle that enables this remarkable defense is called the orbicularis paniculi, and it acts like a drawstring, tightening to seal the animal inside its spiny fortress. Most predators, confronted with this prickly ball, simply give up and look for easier prey. But the hedgehog's spines serve another crucial function: shock absorption. Hedgehogs are climbers and foragers, often moving through trees and bushes in search of food. When they fall, which happens with some frequency, they land on their spines. The internal structure of these spines is specifically adapted to absorb impact energy. Inside each spine, there are thin walls, or septi, arranged perpendicular to the spine's axis. These create a honeycomb-like structure that compresses when the spine is loaded, dissipating energy that would otherwise be transmitted to the animal's body. Tests have shown that hedgehog spines can absorb impacts from falls of several meters, allowing the animal to survive drops that would seriously injure an unprotected mammal of similar size. Porcupines, by contrast, have evolved quills as offensive weapons rather than passive armor. New World porcupines, found in North and South America, and Old World porcupines, found in Europe, Africa, and Asia, evolved their quills independently, but both use them in similar ways. Porcupine quills are longer than hedgehog spines, ranging from about 4 to 15 centimeters, depending on the species and the location on the body. They're also hollow, but their internal structure is different. Instead of the transverse support walls seen in hedgehog spines, porcupine quills have a foam-like core that makes them lightweight while still maintaining stiffness. The defining feature of porcupine quills is that they detach easily and are covered with microscopic barbs near the tip. When a predator attacks, the porcupine doesn't shoot its quills, despite the common myth. Instead, it turns its back, raises its quills, and if contact is made, the quills easily pull free and become embedded in the attacker's skin. The barbs make the quills extremely difficult to remove. With each movement of the predator's muscles, the quills actually work their way deeper into the flesh, causing increasing pain and potentially serious infection. This is a powerful deterrent. Animals that have experienced an encounter with a porcupine remember it and typically avoid porcupines in the future. The quills aren't uniformly distributed across a porcupine's body. The longest, most heavily barbed quills are on the animal's rump and tail. When threatened, a porcupine will turn its rear toward danger, often backing up to drive its quills into an attacker. Some species also rattle their quills together to create a warning sound. The combination of visual, auditory, and, if necessary, tactile warnings makes the porcupine's defense highly effective. The quills are continually replaced throughout the animal's life, so even if many are lost in an encounter, the porcupine isn't left defenseless. Echidnas, or spiny anteaters, represent yet another independent evolution of spine-based defense. These unusual mammals are monotremes, egg-laying mammals related to the platypus, and they inhabit Australia and New Guinea. Echidna spines are short and thick, similar in appearance to hedgehog spines, but with a different internal structure. Like porcupine quills, echidna spines have a foam-like core, though they're not barbed and don't detach. When threatened, echidnas either curl into a ball, similar to hedgehogs, or, if they're on soft ground, they rapidly dig straight down, burrowing into the earth until only their spines are visible above the surface. Tenrecs, small mammals found primarily in Madagascar, have also independently evolved spines. Some tenrec species have spines that closely resemble those of hedgehogs, so much so that they're sometimes called hedgehog tenrecs, though they're not closely related to true hedgehogs. Other tenrec species have developed specialized quills that they can rattle together to produce sound, using them for communication as well as defense. Young street tenrecs, in particular, have a crest of specialized quills around their necks that they can vibrate at high frequency, creating ultrasonic communication signals that help them maintain contact with their mothers. The fact that spines have evolved independently at least 6 times in mammals, and that they show both similarities and differences in structure and function across these lineages, tells us something important about evolution. Spines represent a readily accessible solution to the problem of predation. Hair, which all mammals possess, can be modified through relatively straightforward genetic changes to become stiffer, thicker, and more protective. The developmental pathway from soft hair to defensive spine doesn't require entirely new structures or completely novel biochemical processes. It's a modification of existing machinery, which is often the way evolution works. The variety of spine designs reflects the different selective pressures faced by different species. Hedgehogs, which frequently fall and need to protect themselves from impact, have evolved spines optimized for shock absorption. Porcupines, which face active predators, have evolved quills designed to inflict pain and create lasting negative associations. Echidnas, which can dig rapidly, have evolved spines that work in concert with their burrowing behavior. Each solution is tailored to its specific context, yet all share the fundamental principle of using modified hairs to create a defensive barrier. Modern research into the mechanical properties of these spines has inspired biomimetic applications. Engineers studying hedgehog spines have gained insights into designing lightweight, impact-resistant structures for protective gear and packaging. The hierarchical internal structure of porcupine quills, with its combination of stiffness and controlled failure modes, has informed the development of new composite materials. Even the barbed tips of porcupine quills have inspired medical innovations, including better surgical staples and adhesives that resist removal until they're meant to be taken out. Every protective adaptation carries costs, and the evolution of shells, armor, and spines involves a constant balancing act between defense and other survival needs. Natural selection doesn't optimize for protection alone. It optimizes for overall reproductive success, which depends on many factors. The ability to find food, to move through the environment, to reproduce, to respond to changing conditions. Heavy armor might save an animal from predators, but if it makes the animal too slow to catch prey or too inflexible to mate successfully, that armor becomes a liability rather than an asset. Energy is perhaps the most fundamental constraint. Building protective structures requires enormous resources. Calcium carbonate, chitin, keratin, and bone all demand raw materials that must be extracted from the environment and assembled through metabolically expensive processes. A mollusk building a thick shell must obtain large quantities of calcium from its surroundings. An arthropod producing a new exoskeleton after molting must synthesize vast amounts of chitin and protein. An armadillo developing its osteoderm-based carapace must dedicate significant resources to bone growth. These materials could otherwise be used for growth, reproduction, or simply maintaining bodily functions. The energetic cost becomes particularly apparent in species that face resource limitations. In nutrient-poor environments, animals may develop thinner shells or less extensive armor than their counterparts in resource-rich environments. Studies of mollusks have shown that individuals living in areas with low calcium availability produce thinner, more fragile shells. They're making the best compromise they can between protection and the need to use their limited resources for other essential functions. Similarly, arthropods that live in environments where food is scarce may delay molting, remaining in their current exoskeleton longer than they would if food were abundant, even if this limits their growth. Mobility represents another significant trade-off. Heavy armor makes movement more difficult and energetically expensive. A turtle carrying its shell must expend more energy to walk a given distance than a similarly sized animal without armor. This limits how quickly turtles can move and affects their ability to escape from some types of predators. While the shell provides excellent protection against crushing and biting, it doesn't help against predators that can simply outrun the turtle. Marine turtles have addressed this through streamlined shells and powerful flippers that make them surprisingly fast swimmers. But terrestrial turtles must accept reduced mobility as the price of their protection. Arthropods face a particularly challenging trade-off during molting. The old exoskeleton must be shed to allow growth, but during the molting process and the period immediately after, when the new exoskeleton is still soft, the animal is extremely vulnerable. Many arthropods time their molts to occur in safer locations, often hiding in burrows or among dense vegetation. Some species synchronize their molts with particular seasons or lunar cycles, potentially reducing individual risk through safety in numbers. But there's no way to eliminate the vulnerability entirely. It's an unavoidable cost of having an exoskeleton that cannot grow incrementally. Breathing presents a specific challenge for animals with rigid body coverings. Most terrestrial vertebrates use their ribs to breathe, expanding and contracting their rib cage to draw air into and expel it from their lungs. But turtles, with their ribs fused into a shell, cannot use this mechanism. They've evolved alternative strategies, using muscles in their limbs and shoulders to change the internal volume of their body cavities, creating the pressure differences needed for breathing. Some aquatic turtles can also absorb oxygen through specialized tissues in their throat and cloaca, supplementing their lung breathing when they're underwater. These are elegant solutions, but they add complexity to the animal's physiology and represent additional energy expenditure. Thermoregulation, the ability to control body temperature, is another area where protective structures can create challenges. Thick shells and heavy armor act as insulation, which can be beneficial in cold environments, but problematic in hot ones. Many armored animals must rely heavily on behavioral thermoregulation. They bask in the sun to warm up or seek shade and water to cool down. They may be most active during particular times of day when temperatures are moderate. These behavioral constraints can affect when and where animals can forage, reproduce, and interact with each other, influencing nearly every aspect of their ecology. Reproduction can be complicated by protective structures. Animals with rigid shells or extensive armor must find ways to achieve physical contact for mating. Male turtles, for instance, have evolved concave plastrons that fit over the curved carapace of females during copulation. Some armored animals have evolved elaborate courtship behaviors that allow them to assess potential mates and coordinate their movements. Female animals that lay eggs must have openings in their armor large enough to pass eggs, which can create vulnerable spots in otherwise complete protection. Yet despite these costs, armor and protective structures have persisted and proliferated throughout evolutionary history. The fact that they've evolved independently so many times, in so many different lineages, using so many different materials and mechanisms, demonstrates that under the right circumstances, the benefits outweigh the costs. For animals facing significant predation pressure, or living in harsh environments where protection from the elements is crucial, investing in armor makes evolutionary sense. The key is finding the right balance, the optimal amount and type of protection for each species' particular lifestyle and environment. Natural selection has explored countless variations on this theme. Some species invest heavily in armor at vulnerable life stages, like juvenile stages, when they're too small to flee effectively. Then reduce their armor as adults, when they're larger and faster. Others maintain heavy armor throughout life, but evolve behaviors that minimize its costs, like efficient foraging strategies that reduce the time spent moving around. Still others have found ways to make their armor multifunctional, using it not just for protection, but for thermoregulation, water storage, or display. The evolution of protective structures continues today, as it has for hundreds of millions of years. In our changing world, with new pressures from habitat loss, climate change, and human activities, species are adapting in real time. Some shell-building marine organisms are evolving mechanisms to cope with ocean acidification. Some insects are developing resistance to predators in urban environments. The arms race between predator and prey, between threat and defense, never truly ends. It simply takes new forms, explores new solutions, continues the endless process of adaptation that has shaped life since its beginning. When we look at the shells, armor, and spines that surround us in the natural world, we're seeing the accumulated wisdom of billions of years of evolutionary experimentation. Each curve of a nautilus shell, each segment of a beetle's exoskeleton, each spine on a hedgehog's back represents a solution that worked, a design that passed the test of natural selection. These structures tell stories of ancient oceans filled with predators, of life's movement onto land, of the endless creativity that emerges when survival demands innovation. The protective adaptations we've explored tonight continue to inspire and inform human technology. Materials scientists study the hierarchical structure of shells to design better ceramics and composites. Engineers examine the shock-absorbing properties of hedgehog spines to improve helmets and protective packaging. Architects look to the geometry of turtle shells for insights into building strong, lightweight domes. The principles that govern how nature builds armor, how it balances strength with flexibility, weight with protection, efficiency with effectiveness, offer lessons that extend far beyond biology. In the modern world, many armored species face new challenges. Shell-building mollusks struggle as ocean chemistry changes. Armadillos expand their range in response to climate shifts, moving into territories where they were previously unable to survive. Turtles, with their slow reproductive rates and specific habitat requirements, face pressures from habitat destruction and the pet trade. Understanding how these animals' protective structures evolved, how they function, and what constraints they impose, helps us predict how species might respond to continuing environmental change, and informs conservation efforts. There's something profoundly satisfying about holding a seashell and understanding the processes that created it. Knowing that specialized cells secreted proteins and minerals in precise patterns, that the shell grew layer by layer at the intersection of living tissue and seawater, that its form reflects millions of years of evolutionary refinement, adds depth to its beauty. Similarly, watching a hedgehog curl into a ball becomes more remarkable when you understand the complex musculature and precisely engineered spines that make that defense possible. Seeing a turtle swim gracefully through water takes on new meaning when you recognize the radical skeletal modifications that allow it to carry its protective shell so efficiently. These protective structures connect us to deep time, to the Cambrian seas where armor first appeared, to the evolutionary innovations that allowed life to expand into new environments and new ecological roles. They remind us that evolution is not an abstract process, but a tangible force that has shaped every aspect of the living world. The shells we find on beaches, the exoskeletons of insects in our gardens, the spines of animals in forests and deserts, all carry evidence of their evolutionary history in their structure. As we bring our exploration to a close, consider the remarkable diversity of solutions that life has found to the universal problem of protection. From calcium carbonate secreted by mollusks to chitin fibers arranged in twisted plywood patterns, from ribs transformed into bony domes to hair modified into defensive spines, evolution has explored countless approaches to keeping soft, vulnerable flesh safe from harm. Each solution reflects the specific challenges faced by its lineage, the particular predators and environmental stresses that shaped its development. Yet all share the fundamental truth that survival often depends on being hard to eat, difficult to damage, resilient in the face of threats. These structures are more than just armor. They're windows into how evolution works, how natural selection shapes form and function, how constraints and trade-offs influence design. They demonstrate that there's rarely a single best solution to any biological problem, only solutions that work well enough in their specific contexts. They show us that evolution is conservative, often building new structures by modifying existing ones rather than starting from scratch. And they reveal that some innovations are so advantageous that they arise repeatedly, independently, in different lineages facing similar challenges. If you've enjoyed this journey through the evolution of shells, armor, and protection, please consider liking this video and subscribing to Bedtime Biology. Your support helps us continue exploring the quiet wonders of the natural world. Feel free to share your thoughts in the comments below. What protective adaptations fascinate you most? Are there particular armored animals you'd like to learn more about? Your suggestions help guide future episodes. For now, as you drift towards sleep, you might think about the invisible armor all around us. The exoskeletons of insects going about their lives unseen, the shells of mollusks on distant shores, the spines of mammals in far-off forests, all of them carrying forward the legacy of those first hard parts that appeared in Cambrian seas, all of them participating in the ongoing story of life's ingenuity, its endless capacity to adapt, to protect, to persist. Sleep well, knowing that across billions of years and countless lineages, life has always found ways to defend itself, to build walls against the world, to endure.